Okay, I think I'm going to get started so that we ensure enough time for the speakers. I'd like to thank you all for joining today for our HIRN webinar. Today's webinar is being sponsored by the Human Islet Research Network. My name is Layla Rouse, and I'm with the Human Islet Research Enhancement Center. I work with Dr. Joyce Nyland and Dr. John Cadiz. We are very excited today to present a webinar on the PanKbase, a centralized knowledge base for the human pancreas of diabetes research. We have 4 speakers today, so we have a very full agenda, and we thank you all for joining. Right now, you will all be muted and your cameras off, but there will be an opportunity to ask questions of the speakers at the end of the webinar. We would like to provide some basic background of what is the HIRN, and the HIRN is an evolving ecosystem. It started in 2014 with a series of consortia. So there were 5 consortia, and each representing a distinct niche, unique area of science, followed by our group as the HIREC, helping to integrate all of the groups together. We have now evolved and we have grown over the past decade to include different research components. And now we have our newest group, which is the PanKbase that you will be hearing about today. In total, we have over 1,000 publications across 11 years. We have had over 800 researchers come in and out of our network. We have 200 plus right now. We have had 109 different institutions and 12 different countries represented within our network. Part of our network is to help share and disseminate information across the community, and we do this many ways, one of which is through our newsletter. So our newsletter provides the opportunity to share information about our HIRN members and their recent publications. We share information about upcoming webinars and meetings like this one that you're hearing about today. And we also like to share information about job opportunities and funding initiatives, which you will also hear about today. We currently have 3 funding initiatives within the HIRN network. The first one I'm only going to just flash right here because you're going to hear more about it in the webinar today. It's through the PanKbase group, and the applications are due on September 2nd. The next one is called a Data Scholars Award, which is run centrally through our group, the HIREC. There is a deadline of November 1st, and there are 2 tiers of funding, so we welcome you to learn more about it. I will drop some information in the chat to learn more. And then the last is a HIRN. It's a gateway investigator initiative that's run through the NIH. There are 3 different groups that you can submit an application to participate with, one of which is HIRN, the other is TrialNet, and the other is TEDdy. So that deadline is March 6, 2026. So we welcome you to look at that deadline as well. And we also have a website in which we use to disseminate information about our network, and that's available at HIRN Network. So again, please note there's only one N, HIRN network.org. And there's a tab right here for funding, which we explain all our different funding initiatives. And we also have a resource browser, which we share information about the network, and one of which, here's our resource browser right here. So this again is open to the community, and you're welcome to view data sets, data models, antibodies, get all sorts of information that's been disseminated through HIRN work. And then we also have a YouTube channel. So this recording today is being recorded. This webinar today is being recorded, and it will be posted on our YouTube channel. So that way you can reference it for later, and also to share the link with your colleagues who were unable to join the call today. So with that, I'd like to remind you of a couple things for today's webinar. As I mentioned, it's going to be recorded. Everybody right now is muted with your cameras off, but there will be an opportunity at the end to ask your questions to the speakers directly. And there will be quite a few great topics discussed about the PanKbase program. And with that, I'm going to pass it to Anna Gloyn, who will be our first speaker today as part of our webinar.
Thank you, Layla. Good morning, everyone from North California. Delighted to be one of 4 members of the PanKbase community representing the team today, and we really hope to introduce you to this platform and really take you, get you involved in improving it, contributing data, so let me kick off with who and what we are. So you could probably get the sense from the fact that we have 4 speakers today that we're not just a team of 1. We are actually a large MPI grant that's jointly led by Carl Galton and Steve Parker. And you can see here many of my colleagues who are the MPIs. We're across multiple institutions, and we have a diverse range of expertise covering platform development, type 1 diabetes, data science, computational biology, islet biology, and expertise in engagement and outreach. And we're a newly funded initiative that's supported by the Human Islet Research Network. And our aim is to develop a platform to provide data, knowledge, and analytical tools on the human pancreas to facilitate research in type 1 diabetes and to use modern data science tools and analysis. So you can see here we're a large team of PIs, but as everybody knows, it's not just the PIs that are critical to the success of a program. The real workers are the wonderful staff, trainees, and postdocs that we have within our teams who are driving the engine across the country. Our team, as you can see here, has expertise in many different areas. We have experts in machine learning, pancreas biology, islet biology, genetics and genomics, platform development, and AI. So across today's talk, you're really going to hear how we're leveraging our expertise as a multi-institutional team with this wonderful team of colleagues to integrate data and to establish a resource that we hope will become a really important tool for all of the islet community. So I think it's fair to say that we have never had so much data, and our current data ecosystem is diverse and ever growing. So if you look here at this wonderful schematic that was put together by Marcela, you can see here the many different consortia, not just based here in the US and in North America, but across Europe as well. You can see all the different data sets and initiatives that have been established over the last 10 to 15 years. And you can see that some of them are really centered around diabetes and the pancreas, those at the center of this sphere, and others touch on diabetes with the types of data that they are collecting. But with all of this wealth of data, there is a real need to make it accessible, to integrate it, and to leverage the information that can be gained from these wonderful resources. So some of the challenges, though, that we're facing as we try to navigate this ecosystem is making sure that data is findable and accessible. And this can be really challenging as well because there is a lack of standardization across these resources. And that's understandable. They've grown organically. They've been set up in different countries, and they've had different primary objectives. And this leads to issues with interoperability, integratability. And I think something that we are really acknowledging now is that a lot of this data just is not ready for artificial intelligence. It's not AI ready. There's also a wealth of information that's out there on the web in unstructured text. Let's take the wonderful catalogue of papers that have been published by the HIRN community as an example. How do we make the most of that information? So with these opportunities, these challenges rather, there come some opportunities. We really want to take advantage of the huge investments that have been made in high quality data generation and to be able to leverage that by integrating it to gain statistical power and to be able to do multimodal data integration. And if we can do this, we can really capitalize on the emerging machine learning and AI-based approaches that are really driven by these large data sets. So why do we need PanKbase? Well, I hope with those couple of introductory slides, I've set up this ecosystem that we're working in that's expanding and the need to be able to curate it, harmonize it, and have standardization of analysis so that we can really make sure that it can be used to its fullest potential. So PanKbase is really setting about to democratize access to the data, to provide our community with a catalogue of highly curated data and to ensure that we link the samples to the rich underlying metadata, to develop a platform that can be used by anybody. Doesn't matter your archetype, it will work for biologists, clinicians, data scientists, and to establish and offer analytical tools that can be run in any computing environment. We're looking to work not just within our own team, but also broadly with the international community to develop robust standards for human pancreas research, and critically to engage with experts and the community to ensure that what we're putting together here serves everybody at large. So we're very early on in our mission, and I really want to stress that. For some of you who were at the HIRN meeting earlier this year, we gave a first taste of our beta version at the HIRN meeting. And those of you who were with us in Chicago at the ADA also got an opportunity to see how our beta version has developed. But we really are in a beta version stage. We're here early on our journey up the mountain. And one of our goals today is not just to show you what we're doing, but really to engage with you and to get you to join us on this journey so that we can reach the top of that mountain together in a way that serves the community optimally. So I'm going to touch on just a couple of things before I hand over to Noel, who is going to start to show you how the PanKbase knowledge base actually works. But before doing that, I want to just give you a little bit of information on what happens behind the platform and some of the work that we've been doing to catalog highly curated data on the pancreas and to link it with the rich underlying metadata so that it can be fully leveraged for analysis. So one of the challenges that we face in our community is that we have lots of different places now where we can access human islets for research in our individual laboratories, or go to resources such as the Alberta Diabetes Islet Core or the fabulous Human Pancreas Analysis Program, so that we can access data from pancreases and islets from individuals with and without diabetes. And if you look across here, the number of donors now is really large. There's 170 characterized donors in HPAP, Alberta Islet Core, over 500. And look at the staggering number of islets that have been distributed and studied from the Integrated Islet Distribution Program. Over 600 from NPOD and over 650 from Prodo. But each of these islet providers is set up in a slightly different way, and there is inconsistent use of RIDs across these resources, and that can make it really difficult if you want to combine data from one platform with another to be able to do that in a meaningful way. We also struggle because the biosamples that have been captured, there's different information recorded on them, and the assays that have been deployed are not identical across programs, and there are differences in the depths of phenotyping and inconsistent reporting of experimental protocols. And this is a headache if you want to really leverage the potential of these wonderful resources. So something we've invested heavily in, leveraging again on the expertise across our group, is to bring this data all together and to put together some tiers of metadata standards that allow the community to work very flexibly and very transparently. And we've really anchored these around the standards that were published in 2018 from Heart and Powers. So you can go in now and you can look at the donors and the data, and you can select the level of certainty you want with the data, with required, desired, and optimal characteristics. And you can find all of this on our metadata and standards page. And if you scan this QR code, it'll take you straight there and you can explore in more detail. So what can you do having put together a really well curated set of data? Well, we can bring it together for not just the donors, but we can use it to generate large single cell integrated maps using data from different resources. We can pull in different types of data, such as islet function. Here's some data showing from the PankDB portal. And we can also include islet EQTLs. And by combining this across multiple resources, we increase power, and that's critical for many of these if we want functional outputs, if we want to use them for biological insight. So we've got over hundreds of donors from PankDB, over 1,000 for IDB, and with time we'll be able to bring in the data from Patrick McDonald's Alberta Diabetes Islet Core. When you start to combine data of this scale, you can then derive knowledge from these resources. You can start to do cell type gene expression. You'll hear a little bit more of this from Noel shortly. You can look at changes in gene expression in relation to phenotypes and disease status. You can identify cell type marker genes, and you can look for co-localized EQTLs with type 1 diabetes risk. And this really is just a taster of the sort of things you can start to do when you combine data across resources. So in terms of what to expect from the team today, I'm going to hand over now to Noel, who's going to give you an introduction into the PanKbase platform. Following that, you're going to hear from Jie Liu about PanKgraph. And then at the end, you'll hear from Marcela, Brissova, who's going to really tell you where we are on the mountain and how we're looking ahead to taking the journey to the top together as a team.
Thanks, Anna. All right, looks like folks can see my screen. So now it's time to dive in and start to give you a tour of our initial offering. And if you want to follow along with me, there's a little QR code right there that you can scan, or you can just type in pankbase.org and you'll find what you see in front of me here. I'm going to do a combination of some slides and a live demo today because I'm in the stable Wi-Fi, fingers crossed, and I think that's the best way to show the resource. So Anna set you up beautifully for what we're going to talk to you through. But what we want to show you is what we built so far and how you might use it. And really be, very humble. And our initial offering is really about thinking about bringing everything we can for the human pancreas that we know about in our community, particularly you all in HIRN, together in one place, harmonized and accessible. That's like the theme of our initial offering. And that's just building the foundation for PanKbase, right? But part of that also includes a place for you to start, right? So our initial offering includes a landing base across the human pancreas, so there's some information. As Anna mentioned, multi-site donor metadata standardized in one place, an integrated resource for that data to visualize it on a cell browser and differential expression, multiple access points for interaction based on your particular level of exposure or interest, whether you're computational or you like to actually visualize it as a biologist, the results, and multiple layers in that. Bringing curated knowledge across the other resources that you all know about in this environment for the human pancreas. And as Jay is going to talk about, a knowledge graph focusing currently on EQTLs. So if you follow along, I'm just going to navigate you a little bit to begin with. The first thing is, this is where you can find everything, as you know, classic in a UI, but we're going to walk through different components right now. As you scroll down the page, what we wanted to bring to the forefront was the resources that are unique to PanKbase, that are not ideally anywhere else. So a couple of them I'm going to go through today, but the things you want to focus on is an integrated map of pancreatic islets, as Anna talked about. PanKgraph, which tends to make assertions based on that data and other data. A data library, if you want to find it, get it, and download it, that's where you go. If you want to peruse what we know about the donors and all the samples, that's in the donor sample metadata catalog resource piece. And finally, differential expression. So those are the signature resources I'm going to talk about today. Scrolling down further on the page, you'll see tools and workflows. If you're more, I just want to dig into the tools. I don't want to see like all the bells and whistles. You can go directly to that. Some of this is repetitive at this point, but it's really more to give you structure of the types of things to expect from PanKbase over the coming years. And we'll also have examples on the landing page over time, but you can see those here as click more. At the bottom of the page, we also thought, given that we're part of HIRN, we want to take a lens where we want to say, like, for the other resources in HIRN and beyond that are related to the pancreas, let's curate a summary about them. So if someone lands on PanKbase, they're like, well, how does this differ or the same as NPOD? We started to get, some sort of curated information about the data, the difference, the specialities that each of these resources has, because that's critical to PanKbase, in my opinion, and to our field, because you can't get everything from one resource. You have to have different layers that may span resources or resources that go deep, right? So that our job here was to just summarize that for our user so they can get a sense of what's going on in other resources. So this was all done with you all as members of these resources to curate and make that accessible. So that's some baseline things for the landing page. But let's talk about the real fun stuff. You're going to come here for the first time. You're going to be like, what is the data in the resource, right? So for that, the donor summary. So this was work done by Liza Brusman. This is summarizing the work that Anna talked about that's been laboriously done by many of you all in your respective labs, but also Marcela and Anna and Ha and all these different folks in the PanKbase team to get you an integrated map with these curated metadata values for all our donors. So if you want to look at that, you can browse that with this tool that allows you to pick from different programs that we have right here. So there's an example. I chose Proto in this particular case, and you can pick and choose whichever program you want. You can also add different groupings based on ethnicity, sex, cause of death, description of diabetes, all the metadata fields we have, and visualize them. I'm highlighting NPOD here, for example. And then there's a host of other plots and ways you can visualize and sort of slice and dice the data based on your questions, right? You have ridgeline plots, heat maps, matrices, scatter plots, PCAs. These are things you're familiar with, but what's nice is they're all in one place. They're all the same data, and they allow you to filter and pick and choose. So that's sort of like to give you a bird's eye view of the donor summary metadata, what's in the resource from a donor perspective. So that's component one. And then this also, the final piece is what assays have been done on these donors and how are we slowly making our way through integrating these all in sort of a useful and informative manner within the resource. So that's step two, right? So this we'll talk a little bit more. Then finally, if you just want to get that data, which you could go to other sites, if you know where it is on Pancreas Atlas or NPOD, you can go directly there, of course, because you may know those. But right now, what we've done here, the data library, put it all together in sort of three major tiers. The top tier is like just the facts, ma'am, right? The donors, the biosamples, the assays on those. You can get information summarized across all those. Then the next layer up is the analytics done to those. The intermediate process files, the things that we do in the processing, the principal analysis results from all the processing and the metadata, and getting it ready for downstream analysis. And finally, workflows to attach to all those so you know exactly how we did it, or if you want to recapitulate it. And then finally, the bottom of the page is things like schemas and user guides and like helping you use all that. So these like build off of each other. So you can search through all these and browse through all of these. I'm not going to spend too much time on this because a lot of these you might have seen other resources, but again, it's all together in one place, harmonized, standardized, and easily searchable and browsable. So that's the highlight there. Now it's the fun part, right? So we got some data together, analyzed in one place, curated, now allow you to visualize it if you're that type of person. So these maps you're about to see are not new to all of you. You've seen these in many other resources. Many resources have beautiful ways of visualizing it. Here, we're just allowing you to visualize exactly the data we have in the way that we felt would be best to present it, right? So there's that, and also the differential expression. Again, not new things, but things that we're bringing together for this unique data set to poise us for all the things Anna mentioned. So with that, I'm going to switch to a live demo. So bear with me, and here we go, because I think it's best to see this. So this is the integrated cell browser. You can get to it from the top of the menu bar or the resources, as I mentioned. But you have three current integrated sets to choose from, things that were iterative over time, and obviously the really nice highlight, which is one with all of the resources that Anna mentioned all together in one place. And those have about 500,000 cells, and those are what I have visualized here. So again, you're used to seeing these. These are your classic UMAP plots where you can search by gene or different cell type, and they can all be highlighted and so on and so forth. Not new to you again, but very useful if you're visualizing this particular data set. Down the page, you'll see the different graphs to look by cell type expression and by cell proportion, depending what you're interested in, and a host of fields that you can select and visualize the different plots by, right? So that allows you to basically play and navigate through this data based on all of the metadata fields we've collected in these samples. And then you have your classic top marker genes by cell type. Here I have ductal cells shown because, we finally have resolution here with the cell type to actually get at these cells. And so here you have that sort of visualized here. And then at the bottom, you get all of the different cell types, the gene and their mean expression and their percent expression. So this is all sort of for you in one place. And you can interact with it. And then again, if you want to go directly to the data, you can do that back through here as well, if you simply want to go and get the data here. Piece two, differential expression. Again, you've probably seen these heat maps and volcano plots before, but what's nice about this is it's interactive, it's fast, it's responsive, and it is all from the same data collected across these different resources. So here you can pick and choose based on what you want to see, your comparison, your selected gene of interest, and you can toggle with the thresholds if you're interested in. But again, it all highlights for you interactively here, and the table populates for you. So here I pre-selected this one gene, where then you can get the differential expression by two different means, and you can also see the different metadata fields we have on that. So again, these visualizations are not novel, but again, they're all in one place, visualized and accessible for you. So those are the big ticket items, right? So PanKbase is new, but we want to make sure that everything is well documented and really clear to you. So for that, if you want to learn, right, you can find everything about the data, metadata also in the menu bar up here. APIs, if you're computationally savvy, everything is an API based. You can pull it and call it if you want to integrate it within your resources. Then you have all the things I mentioned with the resources here. And then if you want to learn some stuff, you've got the team, the people, but one that's really important is funding opportunities. We have a call for some resources to go to, AI and very specific machine learning readiness funding in the future for PanKbase. So check that out. I think applications are due September 2nd, and it's really great. And we hope to work with you. So that's a little plug for that. And then finally, if you just didn't hear that thing that I just like threw across to you, because that's a lot, we have everything available on tutorials, right? So if you want to get a tutorial, nice video of PanKbase, PanKgraph, and a couple of the resources I mentioned today, they're all there in the tutorial section. And also everything's in GitHub, open access, like our code is. So with that, though, I guess one thing that I ask and we ask as a team, right, is you all use resources every day in your life. And I guess I think like we have water demonstrating through the system. If any of you have seen Finding Nemo, this will resonate with you. I think of like, you're probably saying, well, that's great. This is awesome. You have an own pace, but now what? Tell me the cool stuff you're going to do next. So I'm going to tease you a little bit that before we hand off to Jie. So directions for the platform is now that we have all the data in one place. That's great. And hopefully we establish a platform that first thing does first, invites you to help contribute, whether you're a user or you want to share data. This is really important for us. The resource only is good as all the contributions, right? And also they're useful to someone beyond the community in a way that you as a data producer intend. I think that's really important. Another thing we aim to do over time is create a holistic picture of the human pancreas and type 1 diabetes. So to do that, you have a lot of disease agnostic resources like the HCA and HubMap, who are doing incredible jobs to standardize how they cell types, aggregating data on the pancreas from other donors, right? So that's also a resource to pull into PanKbase and to push to those resources so that there's this cross-pollination. And because we work with many of these in other settings, we have that advantage to do that, whether through APIs or visualizations or simply straight up direct links to begin with. But here's the fun stuff. We really want to make this a resource not for the pancreas, but understand how, what we learn about the pancreas contributes to type 1 diabetes, right? So a couple analyses that are coming on in a couple months from now is non-negative matrix factorization, ideally to do the thing, uncover those latent patterns in the diverse data sets, right? Latent factors that contribute to the onset or beta cell progression and things like that. That's one analysis we plan to do. Biomarker discovery, ideally for diabetes and its pre-disease states. Transfer learning to ideally predict types of diabetes, heterogeneity, and other tasks along the way. Models from sequence to function, predicting those functional consequences. Again, classic stuff that you've worked with, understanding what's going on with the sequences and variants, that's classic. But since all of this is complicated, and the world is all abuzz and a flitter with AI, we have a lot of models within our resource to make it simple so that we walk the user through it in a way that's workflow-based. So over the course of the next couple of months, we plan to incorporate like an agentic workflow for you as a user to type in prompts based on prompts we're giving you and information that we're saying like, this is what's in the resource, this is what you can learn, guiding you and actually allowing the resource to flow for you the results that you want to see based on your questions. And that obviously those results will be in the space. So we're hoping to make this platform far more, forward-looking in data science, because that's a remit that I think is unique to PanKbase. But we can't do that without these amazing other resources who do really cool things like aggregate this data, make it accessible to others, and have niche expertise that we can really capitalize on in PanKbase. So that's what we're hoping for. We're just getting started. But with that, I will pass it off to Jie, who will tell you some really excellent things about what we've done so far with our knowledge graph.
Hello, everyone. I'm Jie Liu. I'm from University of Michigan. So our team is building the knowledge graph within PanKbase. First, a brief introduction about what is a knowledge graph. So the graph is just has nodes and edges, and a knowledge graph is just actual data structure. We have nodes. In this situation, it's a gene, and we have the properties or attribute for this particular gene. And we also have edges that depict the relation between the nodes. In this situation, the gene belongs to this gene geo-ontology. So using a graph, we express explicitly its variant is a GWAS hit with T1D, and it's also a EQTL with a gene, and this gene expressed in pancreas tissue. So using this particular knowledge graph, we depict the information we have really efficiently. And why we need a knowledge graph in PanKbase? The first reason is more intuitively representing biomedical knowledges. As the knowledge graph I just showed you, we depict a GWAS signal here, a EQTL signal here, and also the gene expressed in pancreas tissue. But think about without this knowledge graph, we have to use 3 tables to represent the same information. So we believe an integrated way of integrating these knowledges will be a knowledge graph instead of the tables. The second reason is really to link different resources and provide the provenance. This is, I will say, art from Marcela, who drew all these related resources for PanKbase related data resources. And for example, just think about the gene CFTR. There are already a lot of resources depict this gene from different perspectives. At example, they use an ID like this. At PubMed, there is an article with a PubMed ID. This particular article probably talk about the gene CFTR. At the GTEx, they talk about the CFTR expression level across different tissues. At the String, they talk about CFTR intact with another gene. And there's also a gene ontology reference from gene ontology resources. So without this knowledge graph, all this information will be really isolated and can be anywhere. And by using a knowledge graph, we really provide the links to the original resources and also provide provenance. This provenance will provide a really important role. For example, when a large language model generates answers, they can rely to this knowledge graph and provide provenance so that to reduce the hallucination of large language models. And also the knowledge graph will really promote FAIR principle. FAIR principle is a data science term, meaning that this data can be findable, accessible, interoperable, and reusable. Just think about this knowledge graph because we have identities, meaning that for this gene, we have a unique global ID, so that we know this type of this sequence of string just represent this gene uniquely. And similarly, we also define schema like metadata. For example, the gene must have a strand. It can be either plus or minus, meaning it's forward or backward, so that this is a required reporting data field in the schema so that we can make sure the data is complete. And here, we also, for this particular edge, this gene belongs to this ontology. This edge has to be predefined, and the function annotation can only go from gene to gene ontology terms. So this is another way of imposing this condition so that the data is really according to some schema. And also we have a collection of ontology terms in the knowledge graph so that we can standardize all these terms. At this moment, we have 0.2 million nodes, 1.6 edges, and it's also growing. Right now, we cover the basic knowledge in type 1 diabetes and islet biology. We have high quality data and knowledge contributed by the PanKbase team, as Anna and Noel already described. PanKbase team is really spending a lot of effort curating the data, QC, and generate this high quality data set. For example, the EQTL, the single cell attack-seq, single cell RNA-seq across different tissues, pancreas tissues, different cell types, and there are more coming in future years. And you also heard there's over 1,000 papers from her literature. So right now we systematically extract knowledge from these 1,000 papers, and in the future we can even go beyond current literature, can go to the entire PubMed that is relevant to type 1 diabetes. We have annotations from other repository and consortia. We have links to other resources both within PanKbase and outside PanKbase. For example, we can easily link to the entities in the data library. We can refer to the pipelines in the analytical library. We can also refer to the internal resources like example and the PubMed. I will not do the live demo, but you are welcome to visit the PanKgraph.org to visit the current set. Right now, we only support 4 questions, but I'm sure the question will increase, and we will use more LLM-supported flexible user interface for question answering in the future. Now, among the 4 questions, suppose I choose the second one, which SNP serve as the lead QTL for a particular gene. So in this situation, after you click here, it will pop into this interface with SNP serve the lead QTL for a particular gene. So here you can tap your gene ID or gene name, and it will let you to choose a gene. For example, here, if I choose the gene CFTR, that will prompt to this particular intermediate page. So here, the question is, which SNP serve as the lead QTL for CFTR? There are 4 categories of QTLs from the current PanKbase resource: either pancreatic eQTL, islet eQTL, pancreatic splicing QTL, or islet exon QTL. There are different types of QTL pipeline in the system. So according to the current situation, we have 3 pancreatic splicing QTL available, and that there are 3 credible sets. If I choose the last one, since there are more, so I click here, it will pop into this particular lead SNP RS7784156. So here, we use AI to generate the answer using according to what available from the knowledge graph. Essentially, we have a graph viewer that is super helpful to show you in the knowledge graph what is available for this particular SNP, and the gene is an eQTL, and there's other genes and QTLs available. And there are also references, empirical available. And you cannot easily get an answer from the web portal, but instead you can write down this cipher, or you can use this API and execute it, and you can get the answer. There are 194 variant about the 3 genes. For future work, we will continue adding new modalities, especially related to immunology, and we will facilitate interoperation with other knowledge graphs. We will build this PanKgraph as open knowledge graph so that anyone can rebuild the same knowledge graph with a shared data loader and ontology and metadata. We will support LLM, agentic AI, hyper generation, and advanced question answering. And most importantly, we really hope the knowledge graph can facilitate new scientific discovery. We will pay a lot of attention to use cases. With that, I think next I will pass to Marcela, who will talk about the future.
So thank you so much. As you heard from Anna, Noel, and Jie, our overarching goal is to build platform based on FAIR principles, platform that will promote open science, reproducible research, data-driven scientific discoveries, especially with the dawn of AI and machine learning approaches and technologies that was recognized by 2 Nobel Prizes awarded last year for chemistry and physics. It is really important to accelerate the translation of basic science discoveries to clinical research and patient care. And we hope that this platform will become destination for diverse research communities, including basic scientists, computational scientists, and clinicians. And our goal is to provide first-in-class education and community outreach to general public and organ procurement organizations, which are critical in supporting diabetes research.
I want to recognize organ donors and their families, without whom our mission would not be possible. So as you heard from Anna, Noel, and Jie, everything starts with metadata and aggregation of data sets across age, sex, different disease states. Recognizing it's important to understand development of autoimmunity, autoantibodies, dysglycemia, and progression to symptomatic type 1 diabetes. And here I would like to recognize work of Dr. Anna Ziegler and her colleagues who focused on vulnerable period in autoimmunity that is associated with early postnatal period. Based on the feedback that we received through our surveys from the community and high interest in genomic data sets, the ones that we started integrating in our platform first were transcriptomic epigenomic data. And in the future, we are hoping to add data sets related to tissue architecture, imaging, to understand spatial relationships of the cells, and overlaying on the top of islet physiology. It was also recognized that it's important to bridge gap between basic science and clinical and translational research. And we are very fortunate to be funded through Office of Data Science Strategy to build collaboration with our colleagues at University of Southern Florida and TEDdy team to build toward common data elements that will connect basic science and clinical and translational research. So when we think about integration and building knowledge that will lead to type 1 diabetes prevention, understanding underlying mechanisms of the disease, we all understand that it starts with genome. We know that type 1 diabetes is complex polygenic disease with a strong HLA component. And although genome is invariant, all other layers of information spanning from epigenome all the way to physiology depend highly on environment, age, and are sex-specific. So by integrating knowledge across these different layers, we hope to bring more understanding into etiology and pathophysiology of type 1 diabetes that will allow us to build better diagnostic tools and therapies, and really building path to understanding causality, building prediction models, and ultimately pave the path to type 1 diabetes prevention. This would not be possible without building partnerships within NIDDK and programs beyond there, really starting within the HIRN and expanding it beyond to initiatives such as NPOT, Alberta Diabetes Institute, Integrated Allo Distribution Program, HubMap, and we are also building international relationships with Human Cell Atlas and their single cell RNA sequencing integration team. So it's really collaboration and partnerships that is going to drive these efforts going forward. And we are hopeful that this will really bring together initiatives within the US and those around the world to bring new data sets and build Pancreas knowledge base going forward. So this is where we are now. We are at the beginning of the mountain climb, and we all know that climbing mountains is really hard, but we do it because it's hard. And so please join us on our journey to the mountaintop. We had assembled external advisory board to have a really good pulse on community needs. This advisory board includes Dr. Todd Brusco with expertise in immunology at University of Florida, Dr. Wei Wang, who has expertise in AI and computational science based at UCLA, Dr. Emily Sims, who is a pediatric endocrinologist with expertise in the clinical research based at Indiana University, and Dr. Marianne Slag-Rupnik, who is a biophysicist, physiologist based at Vienna University. There are many ways you can contribute, and it can start with contributing your data, participating in tool development, annotation, curation of data. And we are hoping that these activities will be supported through opportunity funding. These are our emails that you can reach out to us for questions about opportunity funds, or if you need any other help related to PanKbase activities. So in few minutes that I have left, I would like to tell you a little bit about opportunity funding. Here is the QR code you can scan to look into more information. So this starts, application is due September 2nd. It's a one-year pilot that will start earliest November 15. Research plan, really simple, 3 pages, not including references. And it's important to include plan how you're planning to work with PanKbase team. There are going to be 2 application types with 2 awards for each. One focusing on the partnerships, really building partnerships to develop shared data standards, foster interoperability, and interconnect the needs of different research communities. And then smaller awards focusing on research teams developing AI models to enhance the usability, accessibility, and training capabilities of PanKbase. And with that, I would like to thank you for your kind attention. We are really appreciative of your feedback, and we look forward to hearing from you about how we can make this initiative really accessible to you and useful to you, as you heard from Anna, Noel, and Jay. So thank you so much, and we look forward to your questions.
Thank you so much for presenting. This was a great presentation, and I learned a lot today. At this time, I'd like to ask our 4 speakers to turn their cameras back on, and I'd like to invite anybody that's on this call that has a question that they would directly like to ask the speakers to please use your raise hand function. I saw one person pop off, but it looks like their hand went down. Isabella, I see your hand is raised. Unmute yourself and turn on your camera and ask your question directly.
Yes, hello. Nice
Isabella,
presentation.
hi,
Yes.
thank you.
Hi, thank you. Yeah, I have a question. So, are there any plans to expand the PanKbase to include a spatial transcriptomic data sets like Visium, Visium HD, Xenium, or Cosmix?
May I take this question?
Go for
Yes,
it.
I think that's the goal. As I was alluding earlier, the goal is to bring in spatial data sets related to tissue architecture, and spatial transcriptomics would be one of them.
Okay, great. Thank you.
Okay. Did we get that answered? I'm sorry, my Zoom glitched. Does anybody else have any questions for our speakers? Joyce Nyland, I saw your hand raised. You are on mute. I'm sorry, you're still muted. Let me unmute, see if that will work. Nellie, can you unmute? I don't have the host functionality. There we go. Is that working?
Yeah.
While we're waiting for that, Ava Fain, you have a question?
Actually, I put it in there, but I also wanted to ask it, if I have some data and I want to send it in, it's possible you already have it, just because I've been at it 9 years looking at type 1 and the crossover of type 2 and then everything in between. I mean, they call it type 3C sometimes. Sometimes it's obesity, and I'm not quite sure how I might share my files with you. I mean, I can put them in the cloud, and maybe people can download them and decide if they're duplicates. Well, think about it. I mean, email is best if someone emailed me back. So thank you.
Sure, Eve, we can do that. We're happy to talk that through. We've done this with you before for the common metabolic disease portal. So I'm happy to talk to you about that.
That's great. Thanks, Noel. Joyce, are you on?
I think I'm off mute now. Can you hear me?
Yes.
Yeah, I had to switch to computer. Thanks. A great presentation, everybody. And thank you for doing the introduction, Layla. That was terrific. I put a quick question in the chat. It wasn't clear from your RFP for the current funding initiatives. Does the partnership initiative need to involve AI as well as the research, or only the research involves AI?
I think potentially both. I think both.
Yes,
The answer?
yes,
Yeah.
yes, absolutely.
Okay, thanks for clarifying. Thank you.
And we do have quite a few questions in the chat, so I'm going to scroll up a little bit, and it looks like the first question that I see that's not addressed is from Kiati Jurdhar. I apologize if I'm mispronouncing your name. You're welcome to unmute yourself and ask your question directly, but it looks like they're looking for some clarification about analyzing cell type distribution while accounting for multiple factors such as age, sex, and the AAB status. Is that possible, and how should this be approached?
I may take this. Yes, I think it is possible, but what I learned from my colleagues who are part of the KBase and who had great amount of expertise in computational approaches. I think I've worked for good number of years with Steve Parker, and what I learned from Steve is that you have to be able to incorporate latent variables, something that Noel was alluding to. And I think latent space to me as a biologist is very abstract, but it's possible, but it requires a lot of computational effort. And this is where KBase is going to help community of biologists, for example, or clinicians who do not normally operate in this space.
And I could just add, I think this is one of the areas where the curation of the data is so important, because as I alluded to at the start, the depth of data that we have both on the donors and on the samples that we work with differs across the resources. So autoantibody status, for example, isn't available on every single donor that we can put into PanKbase. So I think one of the things here we're trying to do with that flexibility with the metadata is empower you to only work with the data sets where the metadata that supports your question are available. So I think here you should be able to go in and select a data set with the underlying metadata for your question.
And if I just may follow up on that, just keeping in mind that data sets that we are bringing in on basic science side are cross-sectional studies, and autoantibodies come and go. And what is really remarkable is building collaborations with clinical and translational research. For example, Teddy team, where they are monitoring autoantibodies, right? So it's a longitudinal cohort, and be thinking about how AI can bridge the two worlds of basic science and clinical research is going to be very powerful.
Great, thank you. There are a few more questions in the chat, but again, I'd like to remind anybody that's live, if you have a question you'd like to ask directly, please raise your hand. Shane Kelly asks, are there tutorials for using the API?
Yes, there are. I put that link in the chat. There's one, it should be up there to Shane's request. Yes. And it's under the data page is APIs. If you go into the page, you'll see a blue button that says open documentation. That brings you the GitHub repo for both PanKbase and PanKgraph.
Great. And then there's a question about option for downloading integrated data sets.
Yes, indeed. So if you go to the integrated cell browser and you see the 3 different integrated data sets we have, there's a list of each of them in the description. To the right, there's a big download button. That's where they are. And if you ask us, because again, we've tested it, but you never know. So like the help email goes directly to us. So it's not like it goes to some inbox somewhere. So if you have any problems, give us a holler.
Thanks, Noel. Thank you. It's always good to have a component to help answers. We love
Hello?
Is that me
Hello?
that went down or?
Yeah, hi.
Hi. Sorry, I
Hi,
think the other had freeze.
yeah, it's freeze. I was just wondering if we have some data sets, I mean, if there are some data in the data sets or the metadata integrating also pharmacological treatments. And I know that we have most of the donors are from type 1, of course. So probably we have a lot of insulin treatments, but they might have some other treatments as well. So what do you know about that?
Great question. Anna or Marcela, you're best positioned to answer that, I think.
Sure. So we do have some clinical data available for some of the donors that are incorporated currently. In terms of being able to access those data, though, we have to be very careful with some of the data that could be identifiable for the donor. So we can apply and get the data into PanKbase, and we can be able to use it behind the screen and the scenes, but it's not always possible to make all of that data freely available. But if you want to access it for your own research, there are other routes to go through to make sure you can get all of that underlying data. So I'll see if Marcela wants to add anything, but just to say some of the data when we get into the clinical space can be a little bit more challenging because of IRB agreements.
I agree with what Anna is saying, and we're kind of finding this out as we're progressing, especially access to genetic information and being able to de-identify donors that way. So I think operating in open space and perhaps registering with the programs that you can access this information more privately, I think it's going to be probably path forward.
Because if they are de-identifiable patients or the samples are not identified, I mean, we don't have any data about the patient, the name of the patient, etc., etc. We should have this data, right?
Not necessarily, no. The whole area of what is data that you're allowed to have from an individual and what is the level that can start to de-identify the individual is quite complex. So in the US, anybody who's done their HIPAA training will know that there are different levels of data that you're kind of allowed to see and the minimum information. We'd be happy to chat with you and support you if you want to reach out to me or Marcela and hopefully we can help you navigate how you can get hold of data that is available, but maybe through a data use agreement that allows you to sign up to ensure that you're not going to try and de-identify an individual. So please feel free to reach out and we can see if we can help you more specifically with your question.
Thank you, Anna.
Great. We have now concluded the hour. I'd like to thank our speakers for joining us today. It sounds like there's a lot going on in PanKbase, and we look forward to learning more about the project over the years and as you guys develop. And they seem like wonderful people to reach out to directly if there's any questions about the system, the website, or data. So thank you again for joining. This webinar has been recorded and will be posted on our YouTube page. An email link will be shared with you in your email, hopefully by the end of this week. Thank you very much.
Thank you very much.
Bye.
Bye-bye.